The overall goal of this procedure is to deliver morpholino oligonucleotides via ventricular injections into the larval caudal fin and knock down genes to assess their function during regeneration. This is accomplished by first anesthetizing the larva and placing them into a groove of the agarose mold. Next, the injection needle is inserted into the heart ventricle. Then, the morpholino solution is injected 4 to 6 times into the ventricle with waiting intervals between pulses to allow clearing of the heart. Finally, the caudal fin is amputated and allowed to regenerate for 3 days. Ultimately, the effect on fin regeneration can then be assessed by measuring the regenerated area of the fin using the line tracing tool of Fiji ImageJ open source software.
The main advantage of this technique over existing methods like transgenesis or mutagenesis is that it allows the knockdown of genes by morpholinos in larval tissues that are not suitable for direct injections.
Using glass capillaries with a 0.75 mm diameter, place one into a needle puller and pull the needle with the following parameters. With watchmaker's tweezers and under a stereoscope with a micrometer eyepiece, break the pulled capillary to produce a 20 micrometer diameter needle. Then, use a lathe with a wetted rubber spinning wheel to sharpen the needle and produce a 20 micrometer bevel. Prepare the morpholino stock by dissolving the lyophilized morpholino in 1x PBS